Hi, I'm Zhana Duren, and thanks for the introduction. I was so enjoying all this presentation and almost forgot myself also need to present. Today I'm gonna continue the theme of a gene regularity network. We're gonna talk about the recent method we developed. So we start from a very simple question: is we have healthy people and diseased people, and in many studies we interested in what genes are differentially expressed between them. There has been many methods. Then a more mechanistic question we may interested is so many genes differentially expressed, who caused their differential expression, how they are differentially expressed. That is the question. And to answer this, we all know it is gene regularity network is the main tool. Once we have the network, we know which gene are differentially expressed. And then all these differentially expressed gene is maybe commonly regulated by a very few number of transcription factors. And then those could be the driver of the disease. So to identify the driver, the network is really important. And so if you look at the individual genes regulation, so in the gene regularity network, there's many things. And here is just part of the gene regularity network, which is transcriptional regulation. So if you look at the individual gene, we have promoters, enhancers, insulators, and many different types of regulatory element. Here we call them as REs. And those are non-coding regions. And transcription factors and proteins will bind to the REs and have a looping to gene's promoter and regulate the target gene. So this is the most simplified version of transcriptional regulatory network. The reality is even more complex than this one. Then if you look at the network, for this audience, I think I don't need to explain too much about the difference between co-expression network and gene regulatory network and protein-protein interaction network. I think I don't need. And then for the transcriptional regulatory network, they have three types of nodes, and thus they have three types of edges. So here the TF, RE, and TG is the three nodes. And the three edges, TF regulate accessibility of the RE, which is, we call it chromatin regulatory network. So it has been discussed in previous talks. And RE regulate gene, which is the cis regulation, it is also discussed. And TF regulate TG is the trans regulation. So for example, in chromatin regulation, the transcription factor is controlling the accessibility of the REs. So we don't have a direct observation of this event in cells, but we have a relevant one, which is the transcription factor ChIP-seq binding. So this ChIP-seq data is a foundation of the regulation. So that means binding is necessary for regulation, but it's not sufficient. And in cis regulation, we have RE to TG. We also have some relevant observation like physical loop between enhancer and gene, like high C data. This is also a foundation. It's necessary, but not sufficient. And then we also could have EQTL data, like association between genotype and gene expression. That is the consequence of the gene regulation. Because of the regulation and because of the genetic variation, we can observe EQTL. That means EQTL is sufficient. Then if there is EQTL, it must be a regulation, but not all regulation will have EQTL because many of the important regularity element is super conserved. They do not have any variation. Anyway, those many observed data, it give us a lot about the regulation, but it's like it's not a perfect ground truth, but we still can use it because it's the best data we can get. So we collect many of those ground truths and try to develop a method and benchmark them. So the challenges in GRN is first one is the transcription factor and the regularity element have interactions. They have nonlinear interactions. They work together to determine the gene expression. And for single gene, they have hundreds of transcription factor and dozens of regularity element work together. So it makes the problem so complex. So Second one is because the system is complex, we need large sample to fit the model. But reality is we do not have large amount of sample available to fit so complex system. And third one is we have some knowledge which is useful for gene regulation, but how to incorporate them is a challenge. Let's say we have motif for the transcription factor bindings. Like we know compared to random prediction, like motif is very useful, it's enriched. But we also cannot restrict the regulation as motif match site because many of the regulation, real binding, it's without have any motif matching. So how to using them is another problem. So to do this, I think currently the one best data we can get is the single cell multi-omic data. So now 10x Genomics have these kits, so it's like easily generated RNA-seq and ATAC-seq on the same cells. Now more and more this type of data is generating, and so how to analyze them to get a better gene regularity network is the problem here. So our group recently developed a method called LINGER, Lifelong Neural Network for Gene Regulation. So now I'm gonna talk about this method. So input of this method is we have transcription factor expression, like a couple hundreds of transcription factor expression, and regularity element accessibility. So that is the one gene, one model. For every gene, we have those features. And then the model is we use a neural network to predict the target gene expression across different cells. And then here, to use the motif matching information, we do not want to directly use that. So we use the second layer of neural network. It was used motif matching information. So each node here is called regulatory modules, which is a weighted sum of different transcription factors and different regulatory elements. So here, when we do the second layer, first layer, so we have a manifold regularization, which is, we already know the TF and RE motif matching information. We take them as a graph, and the Laplacian matrix was generated, and we use the manifold regularization to encourage the transcription factor that have motif matching to those REs have a close connection in the first layers. And then after fitting the model, we use interpretable AI technique, Shapley values, to extract the transcription factor and regularity element contribution to the individual target genes. Then it gives us the transcription factor to TG connection and RE to TG connections. Then we can get the TF to RE connection from the first layer. That if TF and RE is activated in the same neuron, then they may have connections. But here, the data is not enough. So we have about thousands or 10,000 or maybe millions of single cells. Even those numbers is large, but many of those single cells is not really independent. Just only a few number of cell types exist. So it cannot give us much independent conditions. So one way to solve the problem is we pre-train the model on ENCODE data. We collect about hundreds of tissues. It's just hundreds, but like it provides a lot of conditions. And we train the same model, and we save the parameters and feature information in a file. And when user use it, they can download this one and retrain the model based on the single cell data. So this is called lifelong learning. It's one type of, it's a Bayesian style transfer learning, we'll say. Now we have each of the parameters, which is could learn as the prior from the bulk data, and the feature information is giving us the confidence of the estimation. If for some parameter, if your estimation is really accurate in the single cell data, you cannot change it too much. Sometimes for some parameter, if the confidence is not very high, then in the single cell data, this parameter could be modified a lot. So that is a lifelong learning framework. We talk about some benchmarking. So data that we used here is the PBMC data, and also have an in silico mixture for human cell lines. And the ground truth is we talked about before, TFRE, we have ChIP-seq data, and RETG, we have promoter capture high C and single cell QTL, and TFTG, we use knocking down data and ChIP-seq data. And then we use the evaluation matrix, is the AUC, and also the AUPR ratios. Here, by ratio, I mean compared to a random predictor, how much better than a random predictor. So here, TFRE prediction is the method Linger is working much better than other methods. And RETG part, we use high C and single cell EQTL based evaluation. It also works much better than others. And this is TF and TG network. It's also compared to other method and performing much better. So one thing I want to mention is AUPR ratio for the previous method in 2021 GRN benchmarking paper talked about the AUPR ratio of the based on ChIP-seq based validation is about 1.2. That means the accuracy of many methods is only about 20% better than a random predictor. So it is not because those methods is bad, it's just because the problem itself is so complex. And in our benchmarking, we observe similar things. The AUPR ratio of the previous method is about 1.17 to 1.29. It's about 17 to 29% better than a random prediction. Using Linger, we're using the bulk encode data, using deep learning, using Shapley value, all those things together, we can improve the AUPR ratio to 2.25. So that is, for my knowledge, this is the first method we can, on average, we can do better than 2 folds in AUPR ratios. So for this part, we improving this network inference. So summary of the network part, we develop a method called Linger, and which is from single cell multi-omic data. The methodology is use the lifelong learning, it's a patient style transfer learning, and use manifold regularization for the incorporate the knowledge, and we use Shapley value to infer the network. And the result is improved a lot. And we show some results. So one thing is we're asking, a gene expression is mostly determined by transcription factors, or is more controlled by regulatory element. So we can do a test of the Shapley values. Then for many genes, we find their contribution is equally, like half and half, like similar by levels for cis and trans. But we find about 2% of the genes mainly determined by transcription factors, and about 4% of the genes mainly determined by cis regulation. Then what are the difference of cis? Then we check those. And one thing we checked is the selection, natural selection signal across different human populations. We find the cis-determined gene is only 9% of them is highly selected. But in trans-dominant gene, 27% of them is 3 folds of them are more like selected. That means those genes are important for the human survival. It's very important gene. It's mainly trans-dominated. Why this is important is because this is the paper from Jonathan Pritchard group 2 years ago. It's talking about EQTL gene are very low in, not depleted in the, it's depleted in the selected gene, but the GWAS gene is enriched in the selected gene. So there's a systematic bias between EQTL and GWAS. And of course, the EQTL1 is definitely more dominated by cis, otherwise there's no EQTL should be observed. So that means to study the GWAS, we should pay more attention to the trans regulation. Sorry. Then another study is we use the IBD, GWAS, differential expression, and a network together. We find one interesting thing is the differential expressed gene and GWAS gene do not have a very good overlap. But interestingly, both GWAS and differential expressed gene are target of a common transcription factors. then that is interesting because a genetic variation in a gene of cis didn't cause too much expression change, but it caused expression to their siblings. That means there maybe have a feedback mechanism in the disease, but this one is still not very clear. How it was have this type of feedback existing is still not clear. Then we can see that based on the GWAS and their importance of transcription factor, we can have a predict if their target is differentially expressed or not. This one is like 0.71 correlation. Then we go back to the first question I asked, how genes are differentially expressed. Here, the transcription factor in the left panel, it is expressed, but their protein is in the cytoplasm. They didn't go back to the nucleus, so it cannot involve in gene regulation. So this transcription factor, we say, is expressed but not active. And the left panel is transcription factor is expressed and also stimulated and go back to the nucleus and bind to the DNA. Then it's involved in gene regulation. This cannot be directly observed. Then if we can, for each individual, can we estimate the transcription factor activity? If yes, then we can test. So the idea is we can only use maybe one sample or very small number of samples. We have multi-omic data. We can run linger and infer the network. Then for a large cohort or like bulk or single-cell RNA-seq data, we can estimate the transcription factor activity for each individual. And then we can test if their activity is differential between diseased and healthy conditions. So here's one example. That is a very old data, bulk microarray data of AML and controls. And there's FOXN1, it's a positive control. It's supposed to be changed. But if you check differential expression, they are not differentially expressed. Then if we check their activity, transcription factor activity, we see there's a significant activity shift. And that is consistent with the previous knowledge because this gene is a tumor suppressor. And also we validate this in the TCGA data. If we divide the people into two groups, high active and low active, we find a significant survival difference between them. Okay, so it's a summary. We talked about the challenge of GRN, and we talked about linger, and we have application of GWAS and a hidden driver by activity test. And I thank my collaborators, funding, and lab members. Thank you.
Hello, amazing talk. I heard that you can use linger to detect TF activity scores and try to explain how genes are differentially expressed across different cell types or conditions. I'm curious about in a more continuous scenario where the cell state or the condition of the cell change gradually rather than like two clusters. Are you able to use the network to analyze the gradual change of this scenario?
It's a really good question. In principle, it should be, but we never tried that one. Here, what we tried is between different individuals, like healthy people and diseased people, compare, and we see the difference. For the continuous things, we never tried it, but in principle, it should be okay.
Yeah, thank you.
Thanks.
Yes, sir. So thank you for the inspiring talk. So I just have one quick question regarding, I think it's a good idea that you pre-train your model on the bulk data set and sort of fine tune it afterwards using the single cell. But the single cell and bulk data set, they're inherently different, right? So when you're fine tuning that on the single cell, did you make any special tricks or you refine your loss function? If so, can you provide more details?
Yeah. Yes, it's a really good question. Yes, it is. So the data magnet of a single cell and bulk is very different. So there's two things. For the single cells, we really didn't use single cells. We use meta cells, like merging the cells together. Then it's like more similar to bulk. That is the first thing we use. If we directly use single cell, it's like timing is very longer and also the scale is different. Second one is for the loss function, the elastic weight consolidation should use Gaussian distance, Euclidean distance, but here we use cosine similarity because cosine similarity is not sensitive to the scale change. Yes, that is the two main things we use to solve the problem.
Okay, all right, thank you.